All right, the final set of structural characteristics of the cell that we're going to discuss today is going to be the cytoskeleton. Now, the cytoskeleton is primarily responsible for maintaining the cell shape, movement of the cell and organelles, and internal transport. And this is done through the aid of three different types of macromolecular fibers: actin filaments, the thinnest, intermediate filaments, and then finally microtubules. These are going to assemble and disassemble as needed. So the interesting thing about the cytoskeleton is, it performs many of the same functions that your skeleton does. Your skeleton is responsible for your shape. The fact that you look like a human is due in large part to your skeleton. It allows you to move. It also it allows you to move wherever you need to go. Now, the interesting thing about the cell cytoskeleton, on the other hand, is that it assembles and disassembles as needed. So it'd be kind of like if you just sat there like a great big circular blob, and then you saw a sandwich, and you're like, whew, I want a sandwich, and whoop, you grow an arm, reach out, and get that sandwich. And then you slowly, disassemble or take apart your arm in order to bring it back and stuff it into your face. Once the sandwich is in your face, you have no need for an arm anymore, and so you go back to just being a blob. So that's a really neat thing about the cytoskeleton. It doesn't waste any time. It assembles whenever it is needed. It breaks down whenever it is not. So it's dynamic. It's going to respond to environmental changes depending on what is needed. The one thing I want to mention about microtubules are pretty large. They're going to interact with motor molecules, kinesin and dynein, to cause movement of organelles. I'm not going to spend a whole lot of time with the shape and structure of microtubules, but I do recommend that you take a little bit of time to look at microtubules. Look at how they are responsible for transporting vesicles throughout the cell. It's pretty fascinating stuff. This would be a great place to look if you're going to be uploading some extra credit videos. So check out microtubule function and transport throughout the cell. Last thing we're going to mention is cilia and flagella. These are going to be external structures found on the outside of the cell. In eukaryotes, cilia are going to be much shorter than flagella. Both of these are going to be responsible for movement of the cell. Cilia are going to move in coordinated waves, kind of like the oars on a really big ship. They're all going to move in a series together. You're going to find these, a great example of where you're going to find these is going to be in the upper respiratory tract to help filter out dust and debris out of your lungs. Flagella are going to move like a propeller or like a corkscrew. They're going to spin around in a circle, and they're going to help push that cell wherever it needs to go. All right, that's it for this chapter. So, just in summary, we first talked about the cellular level of organization. We talked about cell theory and how all organisms are composed of cells. Cells must come from pre-existing cells, and they must be small in order to maintain a high surface area to volume ratio. Then we talked about prokaryotic cells. We talked about their shape and how they come in three different forms: bacillus, coccus, and spirilla or spirochetes. And that in terms of their structure, they don't really have a membrane-bound nucleus. They will have a cell envelope with a cell membrane, cell wall, glycocalyx. The cytoplasm, where it contains ribosomes, nucleoid, and plasmid. And then finally, the external structures we just mentioned briefly, the flagella, fimbriae, and conjugation pili. Then we went on to talking about eukaryotic cells. We talked about how in structure they are much larger and more complex than prokaryotes. They contain membrane-bound organelles, and they also have transport vesicles that move around via the cytoskeleton. We then broke down what some of those organelles are, starting with the nucleus and the ribosomes, and how the nucleus is surrounded by a nuclear envelope containing nuclear pores that regulate transport between the cytoplasm and nucleoplasm. We then talked about the ribosomes and how they are made inside of the nucleolus within the nucleus. And this is going to be our primary site of protein synthesis from mRNA. Then we went on to the endomembrane system. That contains the endoplasmic reticulum. The rough ER is going to be responsible for synthesizing proteins and carrying them out to, or transporting them to, the Golgi apparatus. The smooth ER, on the other hand, is going to be primarily responsible for lipid synthesis and transport to the Golgi apparatus. The Golgi apparatus is going to package, ship, and prepare those proteins and lipids from the ER and send them throughout the cell or out of the cell, depending depending on their need. The lysosomes are going to be produced by the Golgi apparatus. They're going to contain digestive enzymes to help break down any wastes and clean up any extracellular debris found inside the cell. Then we went on to microbodies and vacuoles. We talked about how microbodies are vesicles with specific metabolic functions. A great example of this would be peroxisomes. We also talked about vacuoles, these large membrane-bound vesicles that are used for storage. Primarily, there's going to be storage water, which also provides turgor pressure in plants, nutrients, waste, and toxins. Then we went on to the energy-related organelles. We talked about chloroplasts and how they use solar energy to photosynthesize macromolecules. Their internal structure includes stroma, thylakoids, and grana. We did not spend a whole lot of time with the structure of chloroplasts. I don't want you to dwell on that. Just note that it's going to come back again in Chapter 7, where we will be spending a lot of time with the structure of chloroplasts. So do be familiar with that. And then we went on to talk about mitochondria, how their primary role is going to be the synthesis of ATP from glucose. This is going to happen inside the internal structures, which include the cristae and matrix. Oh, and then we did talk about the cytoskeleton. I'm sorry, I don't have a summary slide for that, but we just covered it, so hopefully you can go back and play the beginning of this video to see all of that again. If you have any questions, comments, or concerns, please feel free to reach out to me at any time.